Thank you very much. It's a great pleasure to be able to address such a varied and interested audience. For you to come after school from, I understand, from as far as New Hampshire and Rhode Island and throughout the Boston area is a real testimonial to your efforts as science teachers. And I want to question, are you a high school, middle school, elementary, all? All high school. Okay. My son is just starting, he's a freshman in high school doing earth science right now. And they actually just talked about hydrothermal vents. So he got to actually comment on them because I dived down to them. So today, what I'd like to do is introduce you to these deep sea hydrothermal vents as a means, as a teaching tool. Of course, the students typically can't get there on a submarine, but there are many, many venues that you can actually feel like you are diving on the site often in real time if it's piped in. And I'm going to talk about chemosynthesis in contrast to photosynthesis. This is the process that supports these entire ecosystems on the deep sea floor. And in fact, as you'll see, because you're all science teachers, the primary mode of chemosynthesis at these vents is via symbiotic associations between bacteria and invertebrates, and even bacteria and protists and myofauna, all kinds of things. And if you think about it, this is exactly similar to plants, which have captured long ago or formed a symbiosis between photosynthetic bacteria, which ultimately evolved to become the chloroplasts, that is the photosynthetic machinery in these plants. So we look at these symbioses not only for their the remarkable diversity, effects that they have on these ecosystems, but also as a model of the evolution, the origin and evolution of photosynthetic, actually plants and algae. So I'm going to, please ask questions at any time. Okay, first to define symbiosis so that we're on the same page. This is a word that was originally coined by Anton de Berry, who is a lichenologist, a mycologist, and he used it to describe associations that involve two or more differently named organisms. In the original definition, he did not assign mutualism, commensalism, or parasitism, plus or minus benefits. It was just living together. Over time in biology, it has come to mean a mutualism, but in fact, anyone that studies symbiosis realizes that it's very hard over the whole life cycle of the various partners to really assign a net benefit or detriment. So we try to use the bigger definition and then define mutualisms when we are discussing them. Now, these are incredibly widespread. I've already told you within eukaryotic cells, both plants and animals and protists. But remarkably, the modern examples between eukaryotes, that is plants, animals, fungi, and bacteria or archaea are incredibly widespread. It is the norm, not the exception. And one of the main reasons this is that unlike eukaryotes, which are a tiny blip on the evolutionary scale, I always have to say that, including us, bacteria and archaea have evolved pretty much over the evolution, the evolution of life and origin of life over the billions of years. And they can do remarkable metabolisms that eukaryotes cannot. So this includes different energy sources. They can, depending on the organisms, they can use light or chemical energy. And that would be just like plants and you and I, we have to eat chemicals, organic chemicals for food. And they also can use different carbon sources, different organic, whether they have to use organic carbon or inorganic, that would be CO2. And these are referred to as hetero, that is other feeding, or auto, as self-feeding, because you and I cannot take CO2 and assimilate it, at least as we do assimilate some, but not as a net carbon source. So they have this whole mishmash of efforts that they can make, including what we'll be talking about today is this chemosynthesis and feeding. Now, I want to emphasize, and I'll try to remember, the types of symbioses and efforts that I'll be talking about that you can take to your classroom. And one of the most amazing things about symbioses, as I mentioned, is that they are widespread. They are everywhere. You can walk outside in the courtyard, pull up a clover, It's a legume. It has nitrogen-fixing bacteria in its root nodules. You can see the root nodules. They're pink because they have something called leg hemoglobin to facilitate keeping oxygen away from the nitrogen fixation apparatus. And of course, well, you don't necessarily want to bring cows into the lab, but termites are a great example of an incredible microbial consortium that allows termites and in cows to actually live on cellulose, something we cannot degrade. I'm sorry, I'm really dry. So you can see, and this is just two examples, there are bioluminescent bacteria that form symbioses with squid. I mean, this is amazing. It's so that they are downlit to match the sky. They feed at night, and predators can't see them because the bioluminescent bacteria exactly mimic the state of the moon, the state of the clouds, whatever it's. So there are many, many symbioses that take advantage of these unique and varied metabolic properties of bacteria and archaea. And I would also emphasize that this gives the partners both ecological and evolutionary potential. The organisms can invade and colonize and maintain in habitats that they never would otherwise. One of the great ones, well, not that you'd be taking your students to a desert, but if you're in a desert community like LA or really any desert, you'll see, I didn't know this till I was out in Baja, you'll see an acacia tree or some sort of a legume, which you can tell. They usually have pods, and they usually have, what are these kind of leaves called? The pinnate leaves, pinnate. And you'll see there'll be one tree, and then all the plants, there are plants around it, and then they diminish. And then there'll be another tree with plants around it. And that's because the legumes are fertilizing the soil via the fixed nitrogen into ammonium. They're self-fertilizing. So the examples are just rampant. And of course, lichens are everywhere, which are fungus and algal or cyanobacterial symbiosis. I already mentioned this. This is an old drawing by Ford Doolittle, but we have our own cells to thank bacteria for, with the our mitochondria evolving from so-called alpha proteobacteria, and the chloroplasts evolving from what used to be called blue-green algae or cyanobacteria. Now, I also want to mention, because this is a huge opportunity for students right now, NIH has just started a project. Is there a board on that side? No. And if you look up NIH and the human microbiome, they are at last taking a more ecological perspective on humans, recognizing that you have your own microflora, a unique skin, not unique, pretty common to humans. There was recently a paper in Science by the group Jeff Gordon. They compared panda bears. They did just all kinds of organisms, and there's a general group that's the same. Your gut, your mouth, your skin. There's two other biomes. But students can look, you can start characterizing your own microbiome. And these are bacteria that are good for you. There are bacteria in the back of your tongue that produce nitrite. This is an antimicrobial. They're keeping the bad guys out. Your gut microflora, when you take antibiotics to take down an infection, you also can suffer losing your own indigenous gut microflora. But it comes back. I mean, it's a symbiosis, co-adaptation, co-evolution, and we're just beginning to explore it beyond what we could culture. And I think Roberto must have talked some about that. Okay, so clearly it's an important process. Now, at the deep sea vents, it was astonishing when these were discovered. I'm sure you've seen them. They're on National Geographic and all. And I've put up, I will get a list of websites. There are a variety of websites that you can go to. One is Ridge, R2K, and one, I can't remember what he calls it, but it's Craig Carey out of University of Delaware. Extreme. I knew I was going, I won't even tell you all the words that were going through my mind, but so it's Extreme. He has a good one, and he's at sea right now on the 9 degrees north and Guaymas Basin. So I'm not sure if they have live feed, but that's just a couple. Anyway, these ecosystems were discovered. It was typically thought that life in the deep sea effectively looked like this, only black. Very, very low biomass, because the surface photosynthetic food would all be decomposed by the time it got down. So when geologists and chemists went out to look for these hydrothermal vents with Elven, they were astonished to discover these this is solid mussels right here, these giant tube worms up to 2 meters in height. There's sea anemones, there's shrimp. It's just abundant life. And it was all clustered around these hot springs. And it totally changed our vision of life in the deep sea. And in fact, what could actually support life. Because effectively, this is geothermal energy converted to chemical energy that's supporting primary production. And I'll tell you, the biomass of these vents rivals that of a rainforest. The tube worms, we know how fast they grow because there have been eruptions. These are, I'll talk about vents a little more, but we watch them where there's just solid lava, and within a year you have a tube worm this tall. So I mean, it's extremely rapid growth, all based on chemosynthesis. And as I mentioned, astonishingly, all of the major macrofauna at the vents have these bacteria as symbionts, whether they're epibionts on the surface or endosymbionts and intracellular. So it just really shook up the scientific world. So I will bring home chemosynthesis again. This is basically the equivalent of a plant, but using sulfur in our case instead of sunlight. There are other physiological groups that can use iron, ammonium. Methane is a kind of a specialized group. But the point is that you basically have a mineral media that you can grow things from scratch. And not only did the discovery of the vents really revolutionize our thinking about our own planet, but also about life on other planetary bodies. In the past, well, in the way past, we always thought about little green men on Mars, and I actually don't know what color the women are on Venus. We then went to, if you were thinking about a carbon-based, water-based life, everybody went to photosynthesis because there's abundant sunlight. But when space exploration got more advanced, realized that there's no water on the surface, no liquid water on the surface of Mars. Perhaps there might be, but at least at this point. Europa is covered by ice, but has ice flows that look like there's hydrothermal activity underneath. All of a sudden, NASA and the planetary scientists had to start thinking about chemosynthesis. Because if life required water, all of the organisms would be in the subsurface. And so finding this on our own planet has really shaken up the planetary studies as well. Okay. Now, in the jargon of what I was using for chemical and carbon metabolism, the organisms that we're going to be talking about, I'll be talking about, they are sulfur chemoautotrophs. So they use sulfur compounds such as hydrogen sulfide, elemental sulfur, thiosulfate as an energy source. They use oxygen as a terminal electron acceptor, just like we do. And then the key for their carbon metabolism is that they can convert single carbon compounds, that is CO2, to C3 moiety. Yep?
I have a question. I mean, if they still use O2 as a terminal electron acceptor, don't they still depend to some extent on photosynthesis?
Yes. The primary production is via chemosynthesis, but yes, but there are other whole groups of chemosynthetic organisms, such as those that you might speculate would be on Mars, such as methanogens that use hydrogen and CO2 as an electron acceptor. So there are some that are technically not associated with photosynthesis, but that's a good point, yes. This would run for a long time if there were a solid nuclear winter, but wouldn't go forever. But you can see why, if you were a plain old animal, that you might want to harness these kinds of organisms, this kind of process, as a source of nutrition. So I'm going to just touch on some of these questions today. I just wanted you to know what kinds of things we are interested in and have been covering over the years. One, and I'll be showing you how widespread they are in nature that we know now, and challenging you and your students to discover new symbioses, because we know they exist and the hints are out there, but it's going to take some digging and some exploring. I should point out that in the case of these symbioses, just like chloroplasts and mitochondria, the bacteria have never been cultured. They've been described for over 200 species based on microscopy, physiology, molecular biology, but have not yet been cultured. So one of the questions I'll be touching on in terms of genomics is, have they started the evolution all the way from two separate organisms, the bacterium, to an organelle that has given up a considerable portion of their DNA. We've been able to address their metabolic diversity, their phylogenetic diversity, and their species specificity. As I mentioned at the vents, I won't be talking about this too much, except that they are the major source of carbon fixation, and I just mentioned with the organelles. So today I'm going to focus on introduce the vents in a little more detail, the diversity of these hosts and symbioses briefly, and then genomics and biogeography, both of which are studies that you can do in your laboratory. Assuming, how many of you have access to DNA sequencing? So that should be, I don't know if Tara or Susan are here. So that should be something that HHMI should provide. And I will lobby them because it is so easy to sequence DNA. You can sequence a bacterial or archaeal genome in 20 minutes. That's not assembled, but you can get all the sequence. And this is very accessible for students. I mean, they do the DNA prep, and obviously they're going to get they'd have to get help with getting the sequencing somewhere, presuming you don't have one yourself. But nobody does anymore. Send it to Korea, send it to Santa Barbara. So anyway, I'll be talking about the genomics and biogeography, both of which rely on sequence analysis. Okay, some background. So, and the students will, all your students love hydrothermal vents. Essentially, these occur where the plates are separating due to seafloor spreading, and new crust is being made as lava wells up. It's parallel to the cone-like volcanoes on land, but in the ocean it manifests itself as a ridge. So this is the so-called axial caldera versus the caldera of a volcano. New lava is welling up, spreading, crystallizing. Along with this, there's a lot of volcanic activity. Fissures open up. Seawater percolates down into the subsurface, interacts with the hot magma, the rocks, and a lot of the chemistry is then geothermally changed completely. And I want to point out, people can look at these when you come up. These are pieces of basalt. So this is, if you've been to Hawaii or any other volcanic habitat, this is lava, very iron-rich, very heavy. The surface, you have to be careful, is obsidian, because when the hot lava hits cold water, it turns into this glassy substrate, and it's very sharp. And so I have a few pieces of that, and then I'll talk about the sulfides in a minute. So, okay, so these vents, and seafloor spreading runs around the world like the seams on a baseball. I mean, quite literally, you can see it's just everywhere. And these were first explored off the coast of the Galapagos Islands and have subsequently been looked at in the eastern, western Pacific. We've been to the Indian Ocean, the so-called triple junction, and all along the mid-Atlantic ridge. Now I say we, I mean collectively, very little of the ocean floor has been explored, but now we're at least getting pinpoints in various parts of the ocean. One of the main vehicles that we use is Elven. I always say the submarine, not the chipmunk, especially this time of year when they start singing. And we also use ROVs, remote operated vehicles, which are tethered to the ship. They're not manned or womaned. It has a pilot and two observers. You can see I guess you can see the porthole right here for the observer, right here. Elven was originally built by the Navy for seafloor observation, primarily looking for unexploded bombs. So the portholes, it's a little bigger than a 6.5-foot diameter sphere. The pilot looks straight out. The observers, though, are at 30 degrees because they assumed everybody would be wanting to look down. With the discovery of 2-meter-long tube worms, you're kind of like looking upside down. A typical dive lasts 8 to 10 hours. So one of my biggest issues, I have no claustrophobia. It's the bathroom, or lack thereof. So there are bottles, which of course men can avail themselves of much more easily. The other little issue is that everything in the sphere is recorded on all of the videotapes. There are many cameras on the outside. And those are open to the public. So I have to say, I gave a lecture to the Harvard alumni once, and a man came up to me afterwards and he goes, I'm a a urologist, have you ever thought about self-catheterization? And I get this package in the mail of all the tubing and everything. I took it to sea just to show the Elven crew. It was pretty funny. So the sub has manipulators, as do the ROVs that the pilots use. We man the cameras and looking out, and there's a basket that changes, the formation changes depending on your samples. Now this is also the slide, I have one more that isn't here, I don't have on this one, but where I do say we have seen much more of the surface of Mars than we have of the ocean floor of our own planet. And do you know why? Sorry, I couldn't hear. Money's one. NASA is a line item on the budget. We have to apply for dive time just with a regular scientific proposal. It's running about $40,000 a day now for the ship. Minute leaves the dock. That's the ship crew, land crew, and the Elven crew. The other one is you can't quite see it. This should be the third picture, kind of this. It's light. Light is attenuated by seawater. At most, you can see about 10 meters. And you've seen the vision of the rovers. I mean, you can see to the whole landscape. So that's an issue. And even Jim Cameron, when he did the film, he brought huge lights down, amazing. But you still could only see not too much further. A good example of this is going to the drive-in theater in the fog. No fog and then fog, because the light comes starting to come in. You're thinking it's the movie, but then it just disappears because water attenuates light. So this is a cartoon of the vents to show you you saw all the animals. They're kind of found around kind of diffuse warm flow vents. Water is percolating down, interacting in the deep subsurface, and then being forced out through fissures. If it's direct, it is about 350 to 400 degrees Celsius. So that's around 700 Fahrenheit. You literally have to be very careful when you're in Elven around these because you can knock over a chimney and fry yourself. And the plexiglass windows, they're this thick. This is about typically 2.5 kilometers depth, so 250 atmospheres. The plexiglass windows start to melt at 80 degrees C. So you're always looking out, being very careful. But most of the animals are found around these kind of almost room temperature, up to 23 degrees Celsius vents, where you have shallow subsurface percolation as well. But what you're setting up here is the gradient that chemosynthetic bacteria need. They're chemicals from this anaerobic, anoxic subsurface, and then oxygen from the overlying seawater. And with the early discoveries, first water samples that came in, they smelled like sulfide, and that was one of the clues that these might be supported by chemosynthesis. And this shows you the comparison between kind of ambient deep seawater, 2 degrees Celsius, just above freezing, whereas vent fluid can be quite high, a pH variation, no sulfide in regular deep seawater, but a lot in the vent fluid. And that's because seawater is 28 millimolar sulphate, which is the most oxidized form, and it's geothermally reduced in the earth and comes up as sulfide. And there are other chemicals, hydrogen, iron, and then oxygen and CO2. So this shows some of the main animals. Unfortunately, my new computer doesn't work, so I can't show you movies. I actually have to mail it back. It's only 5 weeks old. You don't want me near your computers. Here's the so-called one of these black smokers. This is that source fluid venting right out. This is a more diffuse warm vent. There's mussels around there. Here's the tube worms, the giant clams, and then in the mid-Atlantic ridge, shrimp. Shrimp. These are all shrimp. You can see one lone shrimp way up there. Now I wanted to point out that the difference here between the seafloor is that there's the lava, that's the basalt, and then there are these so-called sulfide chimneys. And that's what's formed by this highly acidic, full of metals and sulfide. And when it hits the cold seawater, it precipitates. That's what this black smoke is. And it also forms chimneys and towering spires. And I have some pictures. I have some samples. You can see this one's lined by fool's gold. And you can look at these. And others have, and you can see the tortuous pathways and things close up. Yeah?
So the deep seawater is actually oxygen-free?
No. Deep seawater, oh, Sorry, that should not, no, it's not. It's about half of the saturation of surface seawater. And how that's wrong, sorry. And what happens is it's the cold water at the poles sinks, and then you have that deep circulation. Yeah, sorry, that's wrong, and I don't remember what the saturation level is, but no, so there's quite a bit of oxygen. A lot of people think there isn't. So anyway, you should compare and contrast the sulfides with the basalt. These are two of the substrates. And I mentioned these, you heard some of the giant clams, which actually literally get as big as dinner plates. And I did mention this is 250 atmospheres. We're down in a 1 atmosphere pressurized sphere. One of the fun things to do with your classes, if you know somebody going to sea, is you decorate styrofoam cups and you send them down on the outside of Elven. And this shows what happens under pressure. And it's why you're in a 1 atmosphere, 1 atmosphere sphere. You would be roughly this big. So, and it's a good thing to, yeah, so anyway. Okay, so the tube worms were the first animal in which this symbiosis was discovered. They are unusual in that they completely lack mouth and gut, so they're effectively a plant on the deep sea floor. And I was actually the person who discovered this as a first-year graduate student. We had a lecture that showed that they had sulfur, elemental sulfur in their tissues, and I said, well, it's perfectly clear they must have chemosynthetic bacteria, just like chloroplast. And indeed, we were able to show these are the individual bacteria. They're about 10 to the 11th, 10 to the 12th per gram. So this trophosome tissue that fills their coelomic cavity is just packed with bacteria. They have a specialized blood system that has the hemoglobin is like other invertebrate hemoglobins and related to vertebrate. But it's unusual in that it has an oxygen binding site and a sulfide binding site. So it actually transports the sulfide and oxygen from across this plume, which is the equivalent of a gill or lung, and then taking it down to its bacteria. And of course, I want to emphasize chemosynthesis is not just carbon fixation. These bacteria are providing all of the carbon and energy nutrition for their host. So it's the micronutrients, the macro, nitrogen, everything. Yep.
Now, do these bacteria, do they sort of live in the gut or the remnants of a gut or
No.
intracellular?
No, they are intracellular in this particular symbiosis. It is not the remnant of a gut, and I should know what part of an invertebrate embryo they derive from. My colleague, Monica Bright, if you look up her paper, she's the one that has traced this from from larvae to juveniles. And it's not a gut, and it's a very specialized tissue, though, that's totally devoted to these bacteria. There are mitochondria in it, but it's pretty much solid. It flows like a culture when you cut open a worm. And so this is just comparing the chloroplasts, so they really are like plants. So we do postulate that these are mutualistic symbioses and that the host is benefiting by being able to use non-metazoan carbon and energy sources. The symbiont, in turn, of course, with my bacteriocentric point of view, they benefit from having an animal or something bigger than them, bringing them the energy substrates they need, sulfide from the anoxic portion and oxygen from the aerobic. And these substrates are typically made in these exclusive zones. So if you're a bacterium, and this water is flowing. I said I have to do interpretive dance since I don't have movies. It's an extremely turbulent stochastic environment. If you're a bacterium, these are amphipods, which are much bigger than bacteria, but serve, if they were moving, to explain. You'd be gone in the flow. You can get on a surface. That's one adaptation that a lot of bacteria have. You can move if you're in sediment. We have sulfide below oxygen. But realistically, that niche is very small, except in places like the Black Sea. But so if you can talk some sort of a eukaryote or something bigger than you into bridging this redox, what do we call it, redox environment, the sulfide from below, oxygen from above, then you're golden. And so we would predict, and did, that these should exist widely in nature. And this is the joys for teaching high school biology, is that not only did we discover these events, but they occur in sulfitic sediments. For all of you who've been living in Massachusetts or along the coast, been to a salt marsh or a mud flat, it smells like rotten eggs. In contrast to geothermal reduction of sulfate, there it's biological. So below the oxygenated surface, you have sulfate-reducing bacteria. They're using sulfate as their electron acceptor, turning it into sulfide. That sulfide diffuses up, and these bacteria can use it. So the examples, this is Solomia velum, found along the Atlantic coast. It has the bacteria within its gill epithelial cells. This is an oligocete, a small subfamily. It's actually mouthless and gutless as well. And it has, just below its cuticle, these are where the bacteria are. They're extracellular, but they're inside of a mouthless and gutless worm. This one is amazing because it looks like a rope. But the scale bar is only 100 microns, so 0.1 millimeter. And those are all bacteria on the surface of this worm that have formed this amazing pattern. They have specialized holdfast, they stick. And in these, oh, and then I should tell this one. Well, let me say, this one has a behavioral adaptation. It actually builds a Y-shaped burrow, and it pumps oxygen from above the sediment, sulfide from below. These two, the oligochaetes and the nematodes, migrate up and down in the, through the sulfide oxygen. So they move their bacteria like elevators. And then this last one, this is a colonial ciliate. So you typically think of ciliates as being single-celled organisms. But here they form this colony. And the white is due to elemental sulfur from their bacteria. All the zooids are covered. And they live in mangrove swamps where there are sulfide streams going out into the intertidal, or not intertidal, might be subtidal, but you can see the roots. And they hunker down to get sulfide, and then they come out to get oxygen. And you can actually manipulate these in the lab, so that's how we know this. So this has evolved remarkably widely, and of course I think it's all the bacteria pushing the symbiosis, of course. Now I mentioned for the vents, the dominant macrofauna, among the mollusks, there are a number of different gastropods in the Western Pacific, mussels and clams, and different species depending on the biogeographic province. You can see based on different colors, different species of the tube worms in the Eastern Pacific, and then shrimp in the Indian Ocean and Mid-Atlantic Ridge. The clam, mussel, and tube worms and the gastropods all have intracellular bacteria. The shrimp have epibionts all over their body, and their whole carapace is modified as kind of a chamber to collect the sulfide and oxygen. And they actually have additional appendages that are colonized by bacteria. So remarkable anatomical and behavioral adaptations for these symbioses. Now, I want to mention the ribosomal RNA framework. Did Roberto talk about this? I know some of you may not have been there. Have you worked with this at all? This is just a means of figuring out how bacteria, especially those that can't be cultured, are related to other bacteria. Carl Woese pioneered this, looking across all forms of life. And how many are teaching and came up with that life was not prokaryotes and eukaryotes, that is pre-nucleus and post-nucleus, or with nucleus, but that there are 3 domains. And is this being taught at the high school level? Yes. Hallelujah. I have to tell you, it is an effort to get it taught at this college, let alone others, other than in my own classes. And I always quiz prospective graduate students when they are coming to interview how much, of course they're microbial, so they mostly have had it somewhere. But number 1, they got it in high school. So it's an impressive job. Anyway, it's a good thing because this has reorganized our thinking about how all of life is related. And for symbioses, we have this so-called, well, for any bacteria, we have the so-called ribosomal RNA framework. We can now extract nucleic acids, whether you're doing the direct DNA or what they're expressing, and sequence those genes, and ultimately develop species-specific probes so you can go back to your primary sample and identify the bacteria individually or determine their number. And this has just totally revolutionized environmental microbiology and, in fact, human microbiology, because this is the approach that's being taken to develop, to look at the human microbiome. Now, I brought these to hand out. I want to point out, and that you should point out to your students, and I think maybe Roberto talked about this, that there are so many bacteria, and these are the numbers, and I brought you, at least a good portion of you, your own set of 1 million bacteria. So there are 1 million bacteria, or archaea, per mil of seawater, fresh water, tap water, Belmont Springs water. And it varies a little bit depending on deep sea. Belmont Springs might be even higher. They're certainly bigger and happier and fatter because they're living off plasticizers. And there are 1 billion bacteria, archaea, per gram of sediment or soil. So think about how many bacteria you interact with in a day, and your students do. The bad guys get all the press, and they are in the so far small amount. We are able to culture less than 0.1% of the natural microbial diversity. In humans, in dirt, in soil, on the desktop, we have no idea who they are, except through now, starting with this ribosomal approach and now developing whole genomes out of environmental samples. So getting an idea of the capacity. So this is the approach we've taken. I'm just preaching this because this is something you can do, and we should, I even know places to get grants for sequencing, because it is, there is no one that should not know how to do this, in my humble opinion. Well, I mean for understanding life. So these are some of the, oops, there go 1 million bacteria. So here, you can pass these around. You can take one and pass them. I don't know how far back. It's 1 mil. It is tap water. We have counted them. We know there are 1 million. And I have to say, Cambridge tap water bacteria are big, fat, and happy as well. Sometimes it's, yeah?
So if you only look at, so you just looked at DNA, so you wouldn't know whether these are viable right after chlorination?
No, you can look at RNA so that you know the genes are expressed. And in fact, most of the reasons that it was developed in terms of the, in terms of the probes, you're actually probing the RNA because the cell naturally amplifies RNA up to 10,000 copies in a ribosome. But with other genes, people are working on highly, highly sensitive or fluorescent or detectable beacons that you could actually go after DNA, like single copy or multiple. Yep.
Does this mean you now have a way that you can quantify the bacteria that you can't culture,
Yes. There are 2 advances there. One is you can quantify them because you stain them with epifluorescent stains, use an epifluorescent, and so you can count them. That's how we found out there were so many bacteria and that we were only culturing a tiny part of them. Now there are also molecular methods, quantitative PCR, quantitative hybridization, metagenomes, where you do shotgun sequencing and then can document number of genes. But that's not necessarily the number of types of bacteria. And of course, this sort of genetic information can help direct culture and ways of getting at these organisms. So for us, it's been, so these are some of the types of things we've been able to answer going at it in this way. These are just some of the bacteria at play: mussel, the nematode, the clam, and Solomia. So I'll just talk briefly. This is a phylogenetic tree comparing ribosomal RNA sequences. You basically are looking at gene sequences as a chronometer. There's an ancestor, and as things mutate and things speciate or diverge, it's hard to talk about species of bacteria, you can compare those sequences and ultimately construct effectively a family tree. And this is just focusing on the symbionts within the so-called gamma proteobacteria, which have lots of relatives that interact with plants and animals. It includes E. coli, well, they're in alphas, but agrobacterium and the rhizobia. So a lot of interactions within this group. And as you can see, a number of the symbionts cluster, but they do fall into some of the specific groups. I won't go into detail, but it tells us that this did evolve more than once in nature. I'll show you the complicated tree. You just look at the names. Clearly, some of them share sequences, which implicates they may have picked them up in the same habitats in their far distant past. The mussels and the clams always group, but some of these others jump around, solomyids. So the question is whether they've exchanged bacteria over time or whether they have actually formed the same animals, diverged, and then formed independent symbioses. So there's a lot of different stories here. One of the ones I'm going to tell you about just briefly that is amazing to me, but impacts biogeography, impacts humans having children, and that is how are symbionts transmitted from generation to generation. We know now that in humans, mitochondria are only passed on from the mother, and in most animals, not all. So it turns out in the clams, or in the animals we've been looking at, they fall into so-called vertical, which is parent to offspring. So that's through the gonad or egg, or so-called lateral, that is from outside. Could be two ways, horizontal between contemporaries. An example of that is termites. They're coprophagic. They eat when the baby termites are born, they're sterile, but they eat the other colony members' feces, and that's how they get colonized. And that's actually how human babies are colonized for the large part. Not eating feces, but exposure to the nurses, the doctors, the family, for your gut, et cetera. And then the other option is environmental. And weirdly, the vestimentifrans, among all, and other clams and mussels, but the vestimentifrans have to find their bacteria from the environment. Now, these are mouthless and gutless as adults. So it's an obligate symbiosis for the adult. They have separate sexes. They spawn, release eggs and sperm. Eggs and sperm have to find each other in the dark, get fertilized. Then the larvae has to not just find a vent, somehow it's got to swim its way down in this stochastic environment, and they have to find their bacteria. I really had blinders on. I couldn't believe it, but all of the evidence leaned towards it, and we now have evidence that indeed this is the case, and it's a rather astonishing situation to me. And that's why I was going to talk about genomes, and this influences the genome of the symbiont. This is the parallel, this is the path of becoming an organelle. And in the first intracellular symbiont that we actually sequenced the genome of, we being the Joint Genome Institute, sponsored by the Department of Energy, we did the vesicomyid clam. And I'm just going to tell you briefly as a parallel for organelles. This organism uses sulfur, fixes CO2, and you can see this is the gill. They're found in the gills. The animals pump the sulfide from below, sticking their foot in and actually accumulating sulfide in their blood and oxygen from above. And we worked hard to isolate DNA to sequence this genome. Now, compared to other endosymbioses, which are in insects, which you could look at these bacteria like aphids, things like that, you can squash them and take a look. They have already evolved to become extremely small genomes. The bacteria in these sorts of animals are supplementing the diet, typically particular essential amino acids, because they're living on, is it phloem or xylem? Which one's the new? Phloem? Okay, so P is before a Z. Phloem? Oh, I'm saying it wrong. Phloem? Phloem. Phloem. Okay. Okay, so if they're feeding on xylem, they're really in trouble. So the idea is that you start losing genes. There's a lot of, basically it's so-called Muellers Ratchet, because only one or very few are passed on from generation to generation. So deleterious mutations, loss of genes will accumulate. Well, you'll lose genes, and we know that this is what happens. So with the Ruthia, as we named this symbiont, Ruthia magnifica, it turns out that yes, it's smaller in genome size. It's 1.2 million base pairs compared to the typical insect of only about half that. And the arguments are twofold. One is, well, they haven't evolved based on the fossil record. They haven't been evolving as long. But based on the genome and the function of these bacteria in the host of the nutrition, selection may work that you have to have all of these component genes at this time to assimilate carbon, assimilate nitrogen, and to feed the host. Okay, but the counter-argument to that is, well, chloroplasts started out as free-living cyanobacteria or cyanobacteria-like ancestors, and they've been reduced to a fraction of their genome. They've given up a number of genes to the host, or they've co-opted the host being your nucleus, or plant's DNA. So the other argument is that maybe we're seeing this in a state of transition from evolution of a free-living bacterium through this sort of stage to a true organelle that would have their own complement of DNA but could not replicate on their own. These are just some of the pathways that we found via inference from genomes. And I want to bring this up for your classes because these genomes are available. Virtually every genome is available on the web now for just about every organism. So for comparative genomics, for bioinformatics, for just searching and looking at the divergence between whatever your favorite gene is, this is totally accessible. All you need is a computer. And it's an amazing, amazing database that can be mined for really just about any question. There are people that really don't ever sequence their own DNA. They're just looking at the masses and masses of data and getting at stories that were unheard of even 10 years ago. This is just, we have done comparative analyses now. The Japanese have sequenced a Symbiont, and you can see the genes that are totally shared between the two genomes include their carbon, sulfur, nitrogen, really critically essential nutritional pathways for the Symbiont to provide the host. And I'll just mention that these two genomes are already available. This is in process right now at JGI, and the Riftia genome has been sequenced, kind of. It's so-called metagenome. Because these are environmentally transmitted, you actually get strains. I mean, you could just get a zoo. You could take up anything. But if you look at legumes as an example, peas and clover and, and the trees and all the Acacia, it varies with how specific the host-symbiont relationship is. And this is what we're finding here. Riftia, if you look at just the ribosomal RNA gene as a kind of a name type gene, they're identical. But if you look at more variable regions, they're different. So they're actually taking up more than, and I'm going to talk about that in a bit, but it makes the genome sequencing and the so-called assembly and closing of the genome, putting all the pieces together, much more difficult. And what you end up with is a so-called metagenome. It's of the whole community. And that applies for bacteria in water, bacteria in soil. The more complex the environment, the more complex the metagenome and difficulties in getting it and really understanding it. Okay, I'll talk very briefly about microbial biogeography, because this is another thing that students can do through culture. I mean, you just need media. You can look at variants, for example, of rhizobia. You'll see the study I'm going to talk about. We're doing this in the deep sea using a submarine or an ROV. And I'm sitting there going, this is so hard. Why aren't I doing this in my own, like the Biolab's courtyard? There's rhizobium. In my front yard in Lexington, there's clover there too. So we're actually initiating a project on what I perceive as clover. And luckily, I have a collaborator that will actually be able to tell me, that's not a clover, that's a whatever subspecies. But anyway, the prevailing thinking from, well, even way before 1930s, is that there is no bacterial biogeography. I mean, if you think about animal biogeography, you think of giraffes in Africa and pandas in China and zoos. But bacteria, everything is everywhere. They're so small, they move with currents, they move with the air, that they're just everywhere. And then the environment will select, but everything will be maintained. Being able to get at the metagenomes and being able to amplify genes has completely changed our opinion and the findings about this. And a big impact is this environmental acquisition of bacteria. So we decided to go after the tubeworm symbionts because it became apparent, even to me with my blinders, that they really were getting their bacteria from the environment. And first we decided we wanted to prove that they were out there versus just everything. So we went after them using, because we can't culture them, looking at different habitats and different distances away from tube worms. And we used both settlement devices. This is something that you and your students can do easily. You can put them out in a river. We actually practice on the Charles River. These are pieces of basalt that are in a plastic holder. There are also slides in there. And we incubate them in the environments. I'll tell you the size of the basalt. This is where we came up with the fries. It's kind of like a French fry, and it fits right in a 50 mil cap tube, so you can extract the DNA from it, or the RNA, in its entirety. And then we had to come up with this clever name that went with it, Field Recruitment Isolation Environments, because we've been calling them fries since we started. So this is to go after them both on basalt and to be able to actually look at them with these probes on slides. And we deployed these in Riftia clumps about 10 meters away, or about 1 meter away, and then 10 meters away, and then the so-called off-axis. So this is away from any influence of the sulfide, seeing if there was any difference. And in fact, using the polymerase chain reaction with species-specific primers, we were able to detect them within 1 month and within a year at all sites, from bare basalt near and far, near tube worms in the clump. Verified this by sequence analysis, and also by so-called FISH, fluorescent in situ hybridization, which is, again, using these species-specific probes, but where you're actually labeling. And the green is a universal background. The kind of yellowish is using the Riftia probes. You can see there's a lot of bacteria on these slides, but only a few that light up. But we concluded that these so-called phylotypes do exist in the environment, and the tube worms must find them somehow. We're now moving on to looking at a more variable region, the so-called intragenic ITS. I never can remember what it stands for. Anyway, it's a more variable region that separates two components of the ribosomal RNA. And by going after this, we've been able to look at what goes on in the tube worm. It turns out most of the time there's a single genotype. This is, by the way, this new pyrosequencing, 454 pyrosequencing, where you can do, it's so-called massively parallel sequencing. And you can do thousands, 10 to 100-fold more sequence analysis that you could with based on typical PCR and cloning. And it's revolutionizing our view of microbial diversity as well. It's just incredible. So looking at tube worm, these are the number of reads, so-called reads, that we have. This is a pie diagram of the varieties, typically one in each of these. And in one, there were actually 2 genotypes. We then said, OK, that's what the tube worm is selecting, but is it some sort of an interaction like rhizobia and legumes where there's actually crosstalk between them? And so we looked at these same symbionts using these species-specific primers in the environment. So here we took a water sample where we filtered 200 liters through a 0.4 micron filter with a 1 micron pre-filter. Again, got the same genotypes, 1 and 2, primarily out of the seawater, and this is out of 10,000 reads. We assumed that we would have a zoo, but in fact, it appears that the environment mimics the host, and the question now is, like rhizobium, is the host seeding the environment with senescence, with death, so that you get a preponderance of the phylotypes that you would find in the tube worm? Alternatively, are these bacteria locally adapted to the environment, and therefore the tube worms are taking up what is there? And we are now expanding this survey from about 17 degrees north to 17 degrees south throughout the Eastern Pacific rise. And I'll be able to tell you more later. Yep.
I just want clarification. Does this mean that most tube worms, there's only one kind of symbiont in
Yes.
it? You did thousands of genomes and they were all the same?
Yes.
And one of them you found 2 different genomes?
Yes, and actually, in only one in this study, in an earlier study with 3, but using traditional cloning, we also found the same genotypes, the the two dominants.
And how do the genotypes, like, so you looked at 5 different tube worms?
Yes, this is 5 different, right?
And the 4 that had only one,
right?
Was it the same one in all 4?
Yes, it is exactly the same one. This shows the mutation or a different base pair, and it's only this G compared to these other. And that's based on this ITS, which is one of the most rapidly evolving genes in a bacterial genome. We don't actually know what to look at that's any faster. Because this is, it's not transcribed. It clearly has some sort of evolutionary constraint. It's not random, but yeah. So this was astonishing to me, and this was based between about 800 and 3,000 reads per sample.
And these tube worms all live next to each other?
These were all from the Tika vent site. 3 were from the same clump, and 2 were from areas within a few 10 meters, but not in one clump. Not all in one clump. I didn't tell you the story, but it turns out Monica Bright, you should take a look at a paper in Nature. I don't think I have it here. Let me see. I meant to put her slide in, but I didn't. She actually built baby traps, which of course they don't call them that in the paper. And she got little tiny larvae that had little crevices so that they could get a start. If you look at natural basalt, the grazers just eat them. You never can find them. She'd get them started, and then she would harvest them at 1 year, 6 months, 1 year, whatever time you could get out there. And she's been able to reconstruct. It turns out they do have a transient gut when they're larvae. Between the larval and juvenile stage, they kind of lose it. Everyone assumed that's how the bacteria got in, that it was infecting a modified gut. Turns out, no. The specific bacteria based on fluorescent in situ hybridization come through the body wall. There's a period where they are allowed to come in. They proliferate within the cells. They influence the development of the host, that tissue, the trophosome. And then it's a certain time frame, and then it stops, and there's apoptosis that is cell destruction of all the other cells that get in. So there is very strong selection for these particular strains at a particular time. I still think this seems crazy evolutionarily when you're reliant on these bacteria, but nature is teaching us. So that's the story on that one. And in summary, these symbioses have clearly evolved widely in nature. I do challenge your students to look, I should have, by high school students, not undergraduates, to look for other animals. In the old literature, the 1800s, it's rife with histological descriptions of invertebrates. And if you see granular tissue, often there are bacteria there. It's amazing. The other is I encourage people to look at freshwater environments. I didn't talk about, but there are methane bacteria. So if you have where you see methane bubbling out of streams or lake beds, that's another symbiosis, and I'm dying to find one that's evolved in freshwater. These are nutritional symbioses. We're looking at stages of genome evolution, putatively from a free-living bacterium through to an organelle. And as more and more genomes become available, we'll be able to evaluate that hypothesis. As I mentioned for the biogeography, whether this is an issue of local adaptation, that we're finding what's in the tube worm out of the tube worm, similarly with mussels. And I didn't talk about it, but this has a tremendous impact on free-living diversity. Everything is not everywhere. Think about it. If you have rhizobium, if you have clover, a field of clover, those root nodules are senescing, they're re-inoculating the soil. It's a positive feedback loop. We've seen this with the shrimp epibionts. They run into the sulfides, so those bacteria have an advantage because they're constantly being inoculated. So the free-living community reflects these symbioses. So this is really an unexamined area of biodiversity at this stage. And I've mentioned again and again the free-living aspect to organelles. Our thinking is that if you came back in a few million, I don't really understand animal evolution, but bacterial evolution, maybe longer, that we would actually be looking, and maybe we are right now, and we don't recognize it, sulfoplasts that have lost more of the bacterial characteristics and have already actually become this organelle. And I want to thank you all and encourage you to stay in touch with hydrothermal vent researchers, because we have a number of programs where teachers, in the past, the Rebel program where teachers have come out, and individual teachers as well. So thank you very much, and I'll maybe take one question because it's over. Any questions?
Did you get to name anything after yourself?
You are not allowed to name anything after yourself. Other people have to name things after you, and I was in line for a mussel from the symbiotic mussel. I know, isn't that weird? Who would want to be named after a mussel? But it turned out that a species that they discovered in the at another site that was much more shallow, it turned out to be the same species, so it took that name. So I got bumped, but it's a long, complicated process to name bacteria, and it's only been within the last, I would have to say, 10 years that we can now name uncultured. They used to have them in pure culture, et cetera, but now if you have ultrastructure, physiological, genetic characterization, it's so-called candidatus, and so that's been a big leap forward in understanding in classical bacteriology. So, okay, well, thank you. Let me know if you have any questions.